Okay, we're going to go ahead and get started here now. There we go. And a couple of observations. I got an email from my dean over the weekend that the anatomage tables are en route from California. Those big electronic dissecting tables. That's why I have rearranged the room to allow space for this, and it's supposed to be big. Having done that, they probably won't be here till next year. So they're probably going to get lost in the middle of the country somewhere. So it's like when you order something from Amazon and it goes to Nashville and then back to Memphis, up to Chicago, and eventually trickles its way into Sevierville after 4 or 5 days. So anyway, we are going to continue on the nervous system today. And if you've noticed, I did open up the lab test for muscles. Yeah, I think it's due Saturday or whatever it is. It's the end of the week. I may extend it to Sunday night instead, just to give you a little more time on that. The lecture test on Unit 3 will not open up until the lab test closes. So anticipate the lecture test opening up probably the end of the week. And that'll be on bones and muscles. Now, we are actually into our 4th unit now, so we only have 2 units to go. And the last 2 chapters are very short, about 60 slides each, and that's only about 2 hours worth of lectures. So that's like a lecture and a half of timing. So we are very well on track. We will, today in lab, we will take a look at the special senses. We'll look at taste and smell and the eye. And then next week, we'll look at the ear and the brain, and we'll dissect the eyeball and the brain. So if you want to be on ground and do that, feel free to come in. If not, you can watch me do it up here. I'll be doing it anyway. So other things. If you're going to take AP2 in May term, registration has opened up. If you need to get cleared, let me know. All I need is your W number to clear you. We have 18 slots. We've already filled up 5 of those 18. So I was pretty surprised and that it's starting to fill that rapidly. But if you want to do it, that's great. You'll get an advantage in having it out of the way. You can take micro over the summer. You can take micro in the fall. If that's your plan. So it does free you up for other courses. You get rid of a whole semester in 13 days of lecture. So there's something to think about. It really, I will say, if you're concerned about the pace, the average grade in May term AP2, and I've done this for about 4 years. Well, I've done May term for about 8 years. I've done AP2 for about 4 years. The average grade is somewhere between an A and a B. You're either going to come out of this class with an A or a B. Most people I have more A's in the class than B. So that's all you're doing for essentially about 2.5 weeks is AP2. And it's the pace is no worse than what you would have if you were taking a night class. We cover 1 unit or 1 chapter a night. So, and we get it all done. Anyway, anybody have any questions on anything? The cost of that May term course will be about between $700 and $800. And if you want to take it, and you have questions on financial aid, don't ask us. You'll just have to call Morristown. That's the answer we were given when I asked yesterday about it. So everybody's financial aid is going to be different. But I can tell you that the cost is somewhere between $700 and $800. That includes the hourly rate for the 4-credit course plus fees and associated fees with it. So, which is not bad at all compared to UT or ETSU. Okay, let me get us into the lecture on nervous system, and we were looking, the last time we met, we had talked about the glial cells, the supporting cells for the central nervous system. The central nervous system is the brain and the spinal cord. The other part of our nervous system is the peripheral nervous system. That is where all the nerves come into the spinal cord, all the spinal nerves, all the 30-some spinal nerves have sensory input and motor output, plus the 12 cranial nerves with sensory input and motor output. Those are outside the brain and spinal cord. Now, they have their own glial cells too. They have, instead of 4 glial cells, they just have 2. They have what are known as satellite cells. Satellite cells are the peripheral nervous system version of the astrocytes. If you remember from last week, the astrocytes are the most common, most numerous glial cell that we find in the central nervous system. The astrocytes are shaped like stars, and they hold neurons in place. They support the neurons. They control access to the neuron with glucose and with oxygen. They restrict blood flow so that we don't have toxins go into the neurons of the central nervous system. They help to function as a macrophage if the neuron becomes damaged. They do all that kind of stuff. Well, the satellite cells do the same thing in the peripheral system. They hold the neuron in place. They function as a macrophage. They may limit its access to oxygen and nutrients so that nothing harmful might get in to the neuron. 10 years ago, we didn't know what satellite cells do. 10 years ago, I'd stand up here, and actually probably only about 5 years ago, I'd stand up here and say, well, satellite cells, we know they're there, we don't know what they do. We know they have a function, we just haven't identified it yet. Now we know. They act like astrocytes. The other cells in the peripheral nervous system are the Schwann cells. These are the peripheral nervous system version of the oligodendrocytes. They wrap myelin around the axons of the nerves in the peripheral system. The axons of the neurons that are collected together. They function very much like an oligodendrocyte. They don't wrap as many neurons, as many axons. In the central nervous system, an oligodendrocyte will wrap up to 60 axons around forming a nerve. Only remember, in the central nervous system, we don't call them nerves, we call them tracts. It's the white matter of the brain and the spinal cord are called tracts. Anyway, but here in the peripheral system, we get to call them nerves. And it's about 15 or so axons at a time wrapped by the myelin sheath here. You know, a nerve itself, like let's take the sciatic nerve that runs down from the sciatic, not the greater sciatic notch in your pelvic bone, and it runs all the way down behind your knee and goes down to your great toe. And this this large nerve is about the size of your thumb as it passes down underneath the gluteus maximus. That's why it's such an inviting target for anybody with a syringe that wants to stick you. That's why we don't inject in the gluteus maximus, because this nerve that literally is about as big around as your thumb as it's passing down through here is big. And a nerve is made up of thousands and thousands and thousands of axons. A nerve is not one neuron with one axon. That makes up part of a nerve. But a nerve has lots of axons, and the axons, are clustered, usually in groups of 15 or so, and wrapped by the myelin. The myelin functions as an insulator and a protector. In fact, in the peripheral system, if an axon is damaged, it can regenerate itself. We always say that nerves can't grow back. Well, if the nerve cell is damaged, yes, they can't grow back. But if the axon is damaged, it can repair itself. And the oligodendrocytes will actually wrap like a cast around the broken ends of the axon so they can grow back together. Because if we couldn't do that, how could we ever reattach fingers or hands or arms that are ripped off of the body? How could we transplant any organ if we couldn't get the axons to grow back together? As long as the cell body of the neuron is intact, the axons will regenerate. Now, that's only in the peripheral system and it does not work in the central nervous system. Okay, now let's move on. So let's talk about the neuron. When we talk about the glial cells, the neuron is the functional unit. The neuron does the heavy lifting. It has the ability to respond to stimulus. We like to say that the neuron is irritable. It responds to some sort of stimulus, and it will generate action potentials, and it will transmit action potentials, and it will communicate to other neurons. Neurons will last a lifetime. We don't grow new neurons, at least not yet anyway. What we were born with is what we have now. They generally don't reproduce. Now, there have been some studies, and it's probably going to get stronger and stronger evidence over time that perhaps they do. Perhaps we do see some regeneration in the central nervous system, but nothing conclusive yet. But there's a lot of work being done on that. But we know that our neurons will last a lifetime. Now, neurons can wear out over time, but in our large nerves, in our large tracts, we have so many millions of neurons that if we lose a few hundred or a few thousand, it doesn't matter. There's plenty of neurons there that are still functioning. But generally, they're going to last 80, 90, 100 years. They have a very high demand for oxygen and glucose. They can only live for 5 minutes after if their oxygen supply is cut off, if their glucose supply is cut off. Whereas a muscle cell can last for hours. We talked about this with rigor. You know, a muscle cell can go for up to 12 hours before it's completely dead because it's got plenty of stored oxygen and plenty of glucose available to it, and it'll just keep on contracting. But not with a neuron. A neuron is only good for 5 minutes. Now, every neuron is going to has to have a cell body where its nucleus is located. It will have at least 2 processes. It'll usually have a dendrite and it'll have an axon. The dendrite is the antenna. The dendrite is the receiving side of the neuron. Dendrite receives stimulus. Nerve endings, in the general senses, are those free nerve endings we talked about last week in lab, are nothing but dendrites. They're just a very simple extension of the cytoplasm. They respond to touch and pressure and pain and itch and temperature. Yeah, okay. And stretch. It doesn't, they're just simple little, the end of the nerve, essentially, the receiving end of the nerve. The axon, the other end of the nerve, is where the signal gets sent out. But so every nerve is going to have at least a dendrite and an axon. Now, the cell bodies are very important to us. The cell bodies cluster in 2 different locations in the central nervous system and in the peripheral nervous system. We call clusters of cell bodies nuclei. The cells of the neuron cell bodies, we call them nuclei if they're in the central nervous system. We call them ganglia if they're in the peripheral system. We usually see the ganglia of the cell bodies on the outside of the spinal cord. This is what a neuron looks like, more or less. There's your cell body up at the top. Up there, your dendrites. These are all receiving ends here. And there is our axon. And this is in the peripheral, this is in the central nervous system because we see an oligodendrocyte wrapping myelin around the axon. Now, the advantage of the myelin is that myelin is an insulator that allows us to achieve speeds in the central nervous system of 300 miles an hour, nerve transmission. Now, the longest nerve that we have to send something in the spinal cord is about 18 inches. 18 inches at 300 miles an hour. Or the sciatic nerve in the peripheral nervous system, 300 miles an hour to go, 3 or 4 feet. Can you comprehend? None of us can comprehend moving that rapidly over that short a distance. So for us, it's always instantaneous. So the beauty of this is that the insulation is what allows us to move that rapidly. So we have instantaneous response. And even if we're not insulated, and there are areas of the brain and spinal cord that don't have any myelin, we call that the gray matter, and they're relatively slow. They only travel at 100 miles an hour, but they only have to go about 1 or 2 millimeters. So again, it's instantaneous. When we have a bunch of axons, I guess I'll say a cluster, a bunch of axons, sounds like bananas or something. A cluster of axons together in the central nervous system, we call it a tract. The white matter of our spinal cord is filled with tracts. These are axons of nerves that, they're axons that run up and down to the brain and away from the brain. So the spinal cord in a cross section has a gray area that looks like a butterfly and a white area that surrounds it. And the white area is made up of myelinated axons. We call them tracts. We don't get to call those myelinated axons nerves until we get to the peripheral system. Once we're out of the spinal cord, then those axons are now called nerves. Now the processes are the dendrites and the axons. Now back to this thing about nerves. Consider this. Look at the brain here. We're looking at the optic nerve. Now here's left eyeball, right eyeball. There are the optic nerves here, on the left side and the right. So here comes our optic nerve coming out of the left eyeball. And when it enters the brain, it crosses over with the optic nerve from the other side and they share signals. Once they've done that, we no longer call them the optic nerve. They're now the optic tract. The olfactory nerve does the same thing. We'll look at that in lab this afternoon. The olfactory nerve comes up out of the nasal cavity through the cribriform plate and goes into the olfactory bulb in our frontal lobe. And then the signals travel to the rest, travel through the brain in the olfactory tract. So if it's not in the brain or spinal cord, it's a nerve. If it is in the brain or spinal cord, it's a tract. So dendrites. Dendrites receive information. They get stimulated. They can generate a signal. They become depolarized. They send signals to the central nervous system. The dendrites get all excited and send in signals. They're irritable, but they don't generate an action potential. They generate what's known as a graded potential. A graded potential is similar to an action potential in that it is a depolarization event. But a graded potential won't go very far. Graded potentials run out. Action potentials never run out, but a graded potential does. A graded potential determines if you're going to sense something or not. The ability to sense something depends on perception in the brain. If you can't hear what I say, if I whisper something, she hears it, you don't. As far as you're concerned, it didn't happen. She hears it, okay? So you don't have the perception, she does. What you didn't, because I said it very softly, and it generated a graded potential in her hearing. And that graded potential was a depolarization event that traveled to the cell body in the auditory nerve and was able to trigger an action potential. Yours didn't. Yeah. Or perhaps you had better vision than she does, or you have better vision. You got better vision than she does. Okay, so you can see things at a distance better than you can. You can see things close up better than she can. You can't see anything. So it's all about receiving a graded potential that can trigger an action potential. The only way that we are able to perceive anything is with when we get an action potential. Graded potentials are based on the strength of the stimulus. Graded potentials form based on the strength of the stimulus. If I whisper something, you may or may not hear it. If I tap you with a feather behind your back, you may or may not feel it. If you didn't feel it, your touch receptors were likely being stimulated, but could not generate a graded potential strong enough to form an action potential. Graded potentials are nothing more than localized depolarization events that wear out before they can do anything. And we're glad that we have them, because otherwise we would be overwhelmed by all the sensory input that's affecting us: sound and light and odors and touch and pressure and pain. When you think about, all the things on our skin and all the noises that go on in the background and the odors out in the hall and things like that, we adapt to them, we ignore them.
Otherwise, filter out unnecessary stimuli.
We filter out unnecessary stimuli, and a lot of that stimuli never makes it to the point of awareness, and we're glad for that. So anyway, the dendrites will collect all this information. There's no guarantee it's going to get to the nerve, to the cell body. The dendrites will get stimulated. They will depolarize. The depolarization wave will spread across down the dendrite to the cell body. But if it runs out, nothing happens. You're not aware of it. You didn't hear it, see it, smell it, taste it, feel it, or whatever. You didn't respond to it because the signal wasn't strong enough. Action potentials are different. Action potentials, when they start, don't run out till they're done. They run till they're done. And every action potential is exactly the same, but a graded potential is variable. So there's our dendrites over here on the end of the neuron. We may, in many cases, will have thousands and thousands of dendrites, each one capable of synapsing with an axon terminal, or if it's a nerve ending, being stimulated for touch, pressure, pain, or temperature, any of these things like that. They're going to generate a graded potential that it will go from the dendrites. It has to go, the graded potential has to go here all the way to an area called the Axon Hillock right there. The Axon Hillock is where you have to depolarize the threshold. You have to be able to get that depolarization wave that's coming from the dendrite. If it can hit threshold at the Axon Hillock, it will trigger an action potential. If it didn't hit threshold, nothing happens. Now, the good news is that the resting potential in a neuron is a whole lot less than it is in a muscle cell. It's the resting potential in a neuron is about 70 millivolts, minus 70 millivolts, not minus 90. And threshold's minus 55. So instead of having to go like a muscle cell where you have to go from minus 90 to minus 50 to hit threshold, here, you only have to go from minus 70 to minus 55. You only have to go 15 millivolts. We're a lot more sensitive to the stimulus in the with the neuron than we are with a muscle cell. Now, the axons originate at the axon hillock and will extend all the way down to a series of axon terminals. And you can have, in a muscle cell, you can have up to 300 axon I mean, in a motor nerve, you can have up to to innervate a muscle cell, you can have up to 300 axon terminals so that you can connect to 300 muscle cells. But some of these neurons can have up to 10,000 axon terminals, each one receiving the action potential. So if we hit threshold at the Axon Hillock, then we're going to send an action potential down the axon, and it's going to branch out and go out to every axon terminal, and every axon terminal will be carrying the action potential that it will then transfer to the next axon in line, because you may connect to another, you may synapse with a dendrite, you may synapse with a muscle or a gland. So we don't always have to go to a muscle cell. But there are the axon terminals right there at the bottom of this. And, you know, our action potential will come down from the Axon Hillock through here, and it will come out at every axon terminal. Now, when we synapse, what are we synapsing with? It may be another dendrite on the other side. It may be a muscle cell. It may be a gland. We don't know. It's not in this example here. But there are in one type of neuron in the central nervous system. In fact, the most common type of neuron will synapse will have up to 10,000 axon terminals. Now, so what do we know about an axon? Well, we know that the axon has axon terminals. It releases neurotransmitters. It is not just acetylcholine it can release epinephrine, norepinephrine, dopamine, serotonin, nitrous oxide, any number. There's like 50 or 60 different neurotransmitters that can be used here. Some of the neurotransmitters are excitatory, like acetylcholine in skeletal muscle, will excite the skeletal muscle. It will open up the sodium channels. Some other neurotransmitters are inhibitory. They shut down the action of the neuron. In fact, in some cases, a neurotransmitter can be excitatory in one area and inhibitory in the other area. Acetylcholine, our old friend from skeletal muscle, is excitatory in skeletal muscle, causes it to contract. In the heart, acetylcholine causes our heart to slow down. Same acetylcholine, different response. The axon, as I said, can communicate with up to 10,000 other axons through all of the axon terminals. And see, this is the beauty of the central nervous system, is that we are drawing information from all these, you know, this axon with 10,000 axon terminals is sending information out to 10,000 other neurons, which is sending it out to 10,000 more. Each one of those is sending it out to 10,000 neurons. And pretty soon you're dealing with millions and millions or even billions of neurons communicating. For us, because for us to perceive something, one neuron can't do it all. You know, if I say if the clock on the wall says it is 11:42, to be able to say that, I have to be able to look at that with my eyes and perceive that as a clock. But then I have to know that there are something called numbers on the clock. I have to know what the numbers mean. I have to know what the time means. I have to know what a wall is and the clock is and all that stuff. And then I have to be able to perceive all that information and say, oh, it's 11:42. I have to be able to count those numbers. It isn't just look at the clock. There's a whole lot of other information that has to be drawn together, including it's 11:42. That means we're about halfway through class. That's okay, good or bad. What are we going to do? Where are we going with this later on? Who knows? So all these things are, you know, rolling around that you have to process too. Now, axons in the central nervous system do not survive if they're damaged. In the peripheral system, we can repair them. but in the central system, we cannot repair them. If they're damaged, they're done. But they do communicate with all the other neurons. 99% of the neurons in the central nervous system are what we call association neurons, designed to communicate with other areas of the brain and spinal cord to process information to give us perception. Decelerate 10,000 axon terminals. Okay. Now things do move up and down in the axon besides just the action potential. We have the mitochondria are going to travel up and down are going to travel down the axon. Enzymes are going to travel down the axon. That's great. Other things will travel up the axon. The viruses can migrate up the axon. Toxins from different bacteria can migrate up the axon and affect the central nervous system. For example, tetanus toxin, Clostridium tetani, the bacteria that causes tetanus, what we used to call lockjaw, induces paralysis in the respiratory muscles and in the masseter and the temporalis muscle here. So your mouth is closed and you can't chew, can't swallow. Clostridium tetani is an anaerobic bacteria that produces a poison, a toxin, that damages the muscles and the control of the muscles. And so your patient dies from asphyxiation, lack of food, whatever, in partial paralysis, respiratory system's paralyzed. It's caused by this toxin, this bacterial poison that migrates up the axon to get to the central nervous system, which controls all of our muscles. That's one example. If you've ever had a cold sore, 95% of the population has cold sores. It's called by the herpes simplex 1 virus. Usually manifests itself in this area of your mouth. It's painful, it goes away after a week or 10 days. It is a virus, and the virus inhabits the nerves that are affecting the area around your mouth. Like the orbicularis oris muscle, the nerves that are supplying that. For example, the herpes simplex 1 virus will migrate in there. It doesn't live there because viruses are not alive. Viruses are not a living structure, but the virus will inhabit there. And then when you undergo periods of stress of any kind, then the virus will start replicating. The stress levels cause your virus to replicate. It will manifest itself as inflammation and a sore on one side of the mouth or the other. And then it'll go away because viruses only pop up and go away. They can't exist. A virus is usually, your immune system can respond to a virus in about 10 days. And then, or there's some medications that you can apply to it that are topical that will restrict its time for growth, and it'll go away. Or you do nothing, it'll go away. Herpes simplex 1, very common virus. Most people have it. But it inhabits the nerves, the axons of the nerve inside the cheek, around the area of the mouth. Herpes simplex is classic virus for that. Herpes simplex 2, genital herpes, reacts the same way. Only it doesn't show up around the mouth, it shows up around the external genitalia. Poliovirus. Polio is still a disease that has not been completely eradicated. Rabies virus. When it infects someone or an animal, it moves up through the axons of the animal or the human to the brain. And if not treated in time, it's ultimately fatal. Very few cases of rabies have been survived. And tetanus and herpes simplex. So these viruses and these toxins use the axons as a highway to get into the central nervous system. Now, around these axons, even if they're myelinated, it doesn't protect you from things like this. What the myelin does is it functions as an insulator. The myelin wraps itself around the axon. Myelin is a lipoprotein. It insulates the axon. It increases the speed of transmission. It allows the action potentials to jump down the axon, go from point to point to point, instead of taking its time, like in a skeletal muscle where we depolarize the skeletal muscle and we go depolarize, repolarize, depolarize, repolarize, all the way down the muscle and all the way down into the T tubules. In the axon, it's not important that we run the depolarization wave every step of the way in the axon. We can jump from what these areas called nodes, so we can really pick up the pace of transmission, particularly when we're dealing with these long nerves. Of course, at 300 miles an hour, who really cares? So the myelin is an insulator. We can approach speeds of 300 miles an hour. The unmyelinated axons are going to appear as gray matter. They only transmit at speeds of 100 miles an hour, but the gray matter in our brain is usually the outer 5 millimeters, and so the distances are very, very short. We compensate by having short distances. In the central nervous system, we use the oligodendrocytes to generate the myelin sheath. We can wrap up to 60 axons. We have what's called they call it the sheath gap. It's the classic name is the node of Ranvier. The node of Ranvier looks like this. This is a node of Ranvier here and here and here. These are gaps. The axon is surrounded by a ring of myelin, there's a gap, then another ring of myelin, and there's a gap. And so our action potential, instead of having to run down the axon all the way through every segment of the axon, actually leaps from here to here to here. And it jumps from these nodes of Ranvier, or as they call them now, the myelin sheath gap. In the peripheral nervous system, same thing. We have these clusters of myelin wrapped around the axon. We call the gaps that, they're calling them the myelin sheath gaps. Are still the node of Ranvier. Most anatomists, most healthcare professionals, if they're talking about this, are going to talk about the nodes of Ranvier. It's hard to change, 100 years of anatomy textbooks. So anyway, so the Schwann cells are going to be wrapping the myelin. Now, instead of wrapping around 60, like we see in the central system, we're only going to wrap 15 at a time. Doesn't matter. The myelination is caused by these two cells. And it looks something like this. We wrap the myelin around the axon. It has been described as a Swiss roll, a jelly roll, a pumpkin roll, the way it rolls up on top of it, whatever analogy works. The gaps there are where the action potentials can jump from node to node to node. And it's a special type of nerve conduction so that we can go very fast. And many of our neurons are going to be myelinated. Of all the neurons that we have in the nervous system, we can classify them by structure and by function. Structurally, the most common type of neuron is what's called the multipolar neuron, with many, many dendrites and many, many axon terminals. This is where we have up to 10,000 axon terminals. Most of the 99% of the neurons in the central nervous system are multipolar, meaning they have these many, many axon terminals. Second type of neuron is called the bipolar neuron. Bipolar structure are not the bipolar disorder. It has nothing to do with bipolar disorder. This is structure. The bipolar neuron has one axon and one dendrite. Bipolar neurons show up in only two places. We see them in the eye, in the retina, and we see them in the nose for the olfactory receptors. It's the only place we see these bipolar neurons. They're pretty complicated. Then the other type of neuron that we encounter is the unipolar. It has two axons coming off of the cell body. One axon receives information, one axon sends it to the central nervous system. Most of our other sensory receptors are going to use the unipolar neuron in here. So this sort of gives you an idea of what they look like. There is, on the left-hand side, is the multipolar neuron. You can see it has many dendrites, and it will have many, many more axon terminals in here. This is the number one neuron in the central nervous system. It is 99% of all neurons are multipolar. The bipolar neuron, an axon and a dendrite. You can see right here. This is what it looks like in the retina. There's the cell body. There's the axon with its axon terminals. There's the dendrite with its receptor sites. Over here is the same type of neuron, but found in the olfactory region of the nose. Only the top portion of the nasal cavity is where we have, up in this area, the anterior region, is where we find olfactory reception. So there's our dendrite and our axon. And this is the unipolar. There's our cell body. There's one axon here, and there's the other axon there. One axon receives information, one axon sends information. So we use multipolar neurons for a lot of things. All of our motor neurons, all of our motor nerves are multipolar, because even the smallest motor unit has a nerve connecting to 3 or 4 muscle cells. A large motor unit can connect to 300 or 400, but that's nothing compared to what we can see when we're communicating in the central nervous system, where we can connect to 10,000 neurons. And of course, then we see the bipolar neuron in the retina. And here we see most of our sensory neurons use the unipolar neuron. Now, functionally, that's how they look like in structure. Functionally, we have 3 types of neurons. We have sensory. Sensory neurons are irritated. They send a signal from the sensory receptor to the central nervous system. They are nearly all unipolar, except for the eye and nose. So all of our sensory input, except for vision and sense of smell, are going to be using these unipolar. They're sensory neurons. Motor neurons. Remember that sensory neurons carry information to the brain, to the spinal cord. Motor neurons send information out from the central nervous system to the effector. It can be another neuron. It can be a gland. It can be a muscle cell. It can be cardiac muscle, smooth muscle, or skeletal muscle. And in the big category, what are called the interneurons. You saw what an interneuron looked like last week when we saw the reflex arc. The reflex arc has a sensory receptor that's sending a signal down a sensory neuron into the spinal cord. The interneuron processes the information and sends its response out on the motor neuron to the effector. The interneuron was in the middle of the spinal cord. Well, all of these interneurons are association neurons. 99% of all neurons in our body are association neurons, and they're all multipolar. They are the link between motor and sensory. They're mostly in the central nervous system. They allow us to receive sensory input and then figure out what that sensory input means. Because just because your eyeballs are collecting light rays that are stimulating the retina to generate an action potential doesn't mean you know what you're seeing. The ability to interpret vision takes place all the way back here in the occipital lobe of the brain. Has nothing to do with the eyeball. We have to get that information back here and then figure out what it means. What are we looking at? And what's it mean? Go back to that example of, when you're driving down the road and the light turns yellow, what do? I know what you do, You stop, and that's good. There's nothing wrong with stopping. Okay. Unless you're 300 feet from the light and, there's nobody, it's 3 o'clock in the morning and there's nobody out anywhere and you're stopping 300 feet from the light. That's okay. It's situational because you process all that information. You would just as easily want to stop when you were 30 feet from the light at 2 o'clock in the afternoon with lots of traffic, right? Just your response would be the same. But our situational responses, we have to draw in all this information. First of all, how do you know to stop? Why do you stop? Right. Okay, that's drawn from past experiences and memory. That's why you do it. Otherwise, that thing that's hanging there did something. Am I supposed to do something? Because if you don't have anything to draw on or any previous experiences, what do? Just keep on going. So the interneurons, the association neurons, which represent 99% of all of our neurons, process every bit of information we can to make decisions. So, okay. Now we get to go back and look at how these neurons do it. And they use the same type of mechanism that muscles do. They have channels in there. They have gated channels. And they're going to use sodium and potassium. Neurons have charges on them, just like muscles do. Neurons have leakage channels, just like muscles do. Sodium's always leaking in and potassium's always leaking out. And they have to use the sodium-potassium pump to keep that straight, so that we always keep sodium outside and potassium inside. But they also have gated channels, just like muscles do. They have ligand-gated channels that open up when a neurotransmitter lands on them. And they have voltage-gated channels that open or close when the right charge occurs. Remember, in skeletal muscle, a sodium gate opens up when the charge on the membrane hits -50, and the voltage gate for sodium opens up and sodium rushes into the cell, and it stays open until you get to +30 millivolts. And then when we hit +30 millivolts, the potassium gate opens and the sodium gate shuts. And the potassium gate stays open until you get down to -90 again. These are voltage-gated channels. Chemical channels, neurotransmitter, acetylcholine, sodium channels open up at the motor end plate. But now we get a third category, the mechanical gate. The mechanical gate opens or closes when it gets distorted, when we apply pressure to it or vibration. It can open or close and send a signal based on its opening or closing. Our ability to hear depends on vibration in the inner ear that allows mechanoreceptors, mechanical gates to open up in our inner ear to generate a graded potential that will go to a cell, a neuron in the auditory nerve. If the graded potential is strong enough to trigger an action potential, we hear things because something vibrated that cell. That's a mechanical gate opening up. We deform the gate. You feel something with pressure or pain or touch or vibration because you're deforming the gate. Mechanical gates open up that way, or they close that way. So chemical gates use a neurotransmitter. It isn't going to just be acetylcholine now. We have other neurotransmitters. Voltage channels open up when the membrane potential changes. We depolarize just like we did in muscle cells, only, and we're using sodium and potassium. We don't have to worry about that. We're still using sodium and potassium, and we will depolarize, but now we only have to go from -70 to -55 to get the threshold. Voltage channels open when the membrane charge changes, and mechanical gates open and close when the receptor is deformed. When our inner ear is vibrating, thanks to signals sent from the tympanic membrane, it will open up mechanical gates. When the vibrations stop and we're no longer deforming the receptor, then the gates close. Bless you. Now, we know, just like in a neuron, in a muscle cell, the neuron has a resting membrane potential. The charge on the membrane is about -70 millivolts. It's different from muscle cells. Muscle cells are -90. Neurons are -70. The outside is full of sodium. The outside of the neuron is full of sodium. The inside is full of potassium. The only place you're going to see something different like this in the body is in the heart. The heart uses calcium in place of sodium. Don't, just erase that thought till AP2. So, but we have a positive outside on our neuron and a negative inside based on the presence of sodium outside and potassium inside. It's polarized. We can open up the sodium channels and sodium can come in, and then we can open up the potassium channels and potassium can go out. Usually. There are some other things, depending on the neurotransmitter, it may not do that. So, let's see if this.
Neurons. A neuron, like all cells, has negatively and the human brain alone contains about 100 billion nerve cells called neurons. A neuron, like all cells, has negatively and positively charged ions in the fluids located both inside and outside of the cell. However, the ionic composition of these two fluids is markedly different. The inner surface of the membrane is negatively charged, while the outer surface is positively charged. This unequal distribution of charge creates a voltage across the plasma membrane, known as the membrane potential. In a resting, unstimulated neuron, this membrane potential is known specifically as the resting membrane potential. The value of the resting membrane potential depends on two factors. The first is the presence of concentration gradients across the plasma membrane, especially those gradients for sodium and potassium ions. The second is the permeability of the plasma membrane to sodium and potassium. In the resting state, this permeability depends on the number of non-gated leak channels present in the membrane for these ions. The magnitude of the resting membrane potential, inside relative to outside, is measured in millivolts using a voltmeter and 2 electrodes. We'll now place both electrodes in the extracellular fluid. Note that the voltmeter records 0 voltage. This is because the unequal distribution of charge exists only across the plasma membrane. The intra and extracellular fluids are otherwise electrically neutral. However, when we insert one of the electrodes into the intracellular fluid, a voltage is recorded. The value of the resting membrane potential varies, but on average it is minus 70 millivolts.
Okay, so the charge on our neuron is minus 70 millivolts. We're going to just hang on to that number. That's what is the generally accepted value. Yeah, it fluctuates, but we'll just hold on to minus 70. That's your charge on the membrane. It can change. It can change all the time. We can change the charge when we allow sodium to come into the cell and potassium to go out of the cell. The two types of signals that we can generate here, a change in the charge of the membrane, we can generate a graded potential. Graded potentials, I mentioned them earlier. Graded potentials are short distance depolarization events. They run out. They may or may not reach the cell body of the neuron. A dendrite can become, the end of a dendrite can become depolarized when it's stimulated, but is the stimulus strong enough to last, to go down the dendrite to the cell body of the neuron and reach the axon hillock and still be at, and still maintain a charge at threshold. If we are at threshold, then we'll generate an action potential at the axon hillock. It's the only place where we get an action potential. And if we do get an action potential, it'll run down the length of the axon and run out to every axon terminal. Graded potentials are short-lived. They are dependent on the strength of the stimulus. Action potentials don't care about the strength of the stimulus. All they know is that they're running. Okay. So with a graded potential, we open up an ion channel. Maybe it's a neurotransmitter, maybe it is a voltage channel, maybe it is being deformed as a mechanical gate. But we know that a gate opens up because we've been stimulated and we depolarize the dendrite. Now the question is, how powerful is the stimulus? Is it strong enough to depolarize the membrane of the dendrite so that we can get a charge down at the axon hillock of a threshold? It just depends. Graded potentials can come and start in the dendrites, they can come on the cell bodies, but action potentials only come at the axon hillock. You have to hit threshold. So the only thing that you're going to open up the axon hillock is a voltage channel. You have a sodium gate that opens up at minus 55 millivolts and sodium floods in, and you have an action potential that's going to run. A graded potential is based on a neurotransmitter. It's based on being deformed. So the other two types of gated channels are going to generate the graded potential. And of course, graded potentials are only a local event. Only one dendrite may be involved here. Is it going to get to the axon hillock? But with an action potential, it will run down the axon. It will go to every axon terminal with exactly the same strength, because all action potentials are alike. So we deform a sensory receptor. We touch a touch receptor, or stimulate a pain receptor, or a temperature receptor. We deform it. Or we release a neurotransmitter. You have a axon terminal releasing a neurotransmitter that lands on the dendrite and causes a sodium channel to open or a potassium channel to close. That's where the graded potentials come from. It's either mechanical or chemical. You end up getting a localized depolarization event. That's all it is. Which may or may not have the strength to get to the Axon Hillock. Because it will wear out. The stimulus doesn't last. The action potential will run and run until it's done, but the graded potential will wear away. The further you get away from the stimulus, it's going to wear out. So these things aren't going to last very long. You know, if you can get the graded potential to go from the dendrite to the cell body, that's great. That's only a short distance. To get from the axon hillock to the end of the axon terminals, maybe as great as 3 or 4 feet. It has to run down, like the sciatic nerve has to run the whole length of the nerve. Graded potentials wear out. Graded potentials explain why we are sensitive to some signals and not others. Why we're all different. Why do we all have different levels of hearing and vision and sense of smell and taste? You can taste things that I can't. I can hear things that you can't. You can see things that nobody else does. You know, that was a joke. Okay. There we go. Okay. No, everybody has different levels of awareness. You know, why do people have different types of eyesight? Why do they have different levels of hearing or detecting odors? You know, everybody can smell things differently, can detect them at different levels. If you've ever had a hearing test, you know, and they put the headphones on you and they say, do you hear this? Do you hear this? And they go from the low range to the high range. And pretty soon you're so tired of listening to it that you don't know if you're hearing it or not. You know, you've been there. I know I have. It's like, yeah, sure, whatever, you know. And, but, you know, we find we may be very good at the low end or very good at the high end or very good in the middle, and we don't have any lows or highs, you know, because everybody hears differently. It's the same signal, but it's not generating the graded potential that's forming. Can't generate an action potential. They wear out. But the action potential is going to be different. The action potential, once it starts, it will always run. The graded action potential will run the entire length of the axon. The graded potential is dependent on the stimulus strength. The action potential is not. The action potential doesn't care how strong the stimulus is. It doesn't care if you hit your thumb with a hammer or tapped it with a pen. As long as there's a signal, as long as an action potential gets generated from that nerve cell, it's going to run. You know, it doesn't matter what the source is. And the action potential is going to be exactly the same. The action potential will be the same size, whether you hit your thumb with a hammer or tap it with your phone. Now, how do we know the intensity? Because there's a big difference between hitting your thumb with a hammer and hitting it with your dropping your phone on it. There's a very big difference. The action potentials are all identical, but the rate, the frequency of generation tells you the intensity. You are going to get an action potential exactly the same size when you drop your phone on your finger, as opposed to hitting your finger with a hammer. You're going to get action potentials exactly the same size. But when you hit your finger with the hammer, you're going to generate a lot of action potentials all at once. The frequency of occurrence is going to be very rapid. That's how we can tell it's intense and then it hurts, because we get lots and lots and lots of identical action potentials. When you drop your phone on your finger, you get an action potential, you get an action potential, an action potential. Because the phone's not going to hurt. So the action potentials are all identical, but we measure intensity by the frequency of occurrence. Graded potentials can only respond to the strength of the stimulus. Action potentials don't care. The the speed of the conduction has no bearing on the intensity of the stimulus. You're going to send a, whether you drop a hammer on your finger or you tap your finger with, drop your phone on your finger, the signal is going to get to your brain just as fast. It doesn't matter if it's 100-pound weight on your foot or a feather on your foot. If you've got an action potential, it's going to run just as fast each time. Now, something else is, how long will this signal last? Rated potentials only last as long as the stimulus lasts. If we're sticking a pin into your finger and irritating your finger, it will, as soon as we pull the pin away, it'll stop hurting, hopefully. But action potentials have to reset themselves every time. Action potentials have a what we call a refractory period, where we have to reset the voltage channels. It's a very, very brief period, but every time we generate an action potential, we have to reset the voltage gates each time. We're not aware of it. We don't notice it because it's too fast for us to be aware of. So. And if you want to call the action potential a nerve impulse, that's okay. It means the same thing. Action potentials are a little more precise, but if we say nerve impulse, and if you remember, we started out talking about nerve impulses when we started originally talking about stimulating the muscle cells. The nerve impulse comes down the motor nerve. It's the action potential, same thing. And we know that they work just as well in nerves as they do in muscle cells. Okay. Let's see what we have to say here.
Neurons communicate over long distances by generating and sending electrical signals called action potentials. Most action potentials originate in the axon's initial segment, a region also called the trigger zone.
Axon Hillock.
During an action potential, voltage-gated sodium and potassium channels open and close, thus altering the membrane's permeability to sodium and potassium ions. As these ions diffuse through the channels, the membrane potential changes. It depolarizes from the resting state of -70 millivolts to a peak value of +30 millivolts, and then quickly repolarizes back to -70 millivolts after a brief period of hyperpolarization. In this topic, we explore, step by step, the events that occur as an action potential is generated in the axon's initial segment.
We don't make that little noise though. Okay, so what happens is we have sodium channels and we have potassium channels. Now the sodium channels are going to open up as soon as we hit threshold. Threshold's a little different here. Threshold on the neuron is -55. So it's and the resting potential is -70. It's much easier to get to threshold. When the sodium rushes in, it will depolarize that area of the where the gates are down from -70 to -60 to -50 or to -55, which causes sodium gates next to it to open up when we hit threshold, and the ones next to them open up. Now the sodium rushes in and changes the charge on the membrane, and just like in the muscle cell, it will raise the charge all the way up to +30 millivolts. At that point, the sodium gates shut and the potassium gates open. Now the potassium gates will allow potassium to leave the neuron, go out into the intracellular fluid, and the charge in the membrane will go from +30 back down to -70, but it will keep on going and bottom out at -90 millivolts and then bring itself back up to -70. So we have a little bit of an overshoot We call this is the hyperpolarization, where we make the membrane a little more negative briefly. So when we return back to -70, we keep on going until we get to -90. So at the resting state, everybody's closed. We get we have a localized depolarization that spreads to the Axon Hillock. It's a graded potential. If we hit threshold, then the sodium channels open up. Sodium rushes in, we depolarize that region of the neuron, which spreads to the next region and the next region. The charge changes from -7, from threshold all the way up to +30. The sodium gates close, potassium gates open up. We are now repolarizing. And when we repolarize, we're going to keep the sodium gates open until we get down to not just -70, but all the way down to -90, and then we'll come back up. So the fourth step is the hyperpolarization stage. We get down to, we hyperpolarize, the sodium gates are reset now. The potassium continues to leave the cell, and then we finally get our potassium gate shut, and we are now back to where we were before. But remember, this is repolarization. We're back to where we were before electrically. We're now positive on the outside and negative on the inside like we were at the beginning of this process, except that potassium's outside the cell and sodium's on the inside, just like we had in the muscle cell. So we use the sodium potassium pump to reestablish the concentrations without altering the charge. Once we've gone through a depolarization, repolarization event, we end up with a positive charge again on the outside of the cell and a negative charge on the inside, with sodium outside, potassium inside. Repolarization simply puts the charges back where they belong, even though there is this big undershoot down to -90. So depolarization, sodium going in, you've seen this before. Repolarization, potassium going out. You know, every time when we open up the voltage gate for sodium and we start depolarizing, as soon as we depolarize that region to +30, the sodium gate's shut and the potassium gate's open to repolarize us back down, in this case, to -70. Threshold is -55. Don't forget that. Threshold is -55. However, what tends to always happen in these neurons is we have this curve like this. We tend to see depolarization followed by repolarization to get us back to -70, but we have a dip right here where we go all the way to -90. We undershoot because we tend to allow too much potassium to leave the cell. Now, there's a reason for this. The reason for this is to allow the sodium gates to reset themselves. So when we hyperpolarize, remember what's the resting potential of a neuron? -70 millivolts. So you have to get a graded potential to generate an action potential. A graded potential has to go take the cell membrane of the neuron and depolarize it. Has to have a localized depolarization to get from -70 to -55 to hit threshold, right? But what happens if the resting potential on the membrane isn't -70, it's -90? That graded potential may not have the legs to get you to threshold. So when we are in the undershoot stage here, when we're all the way down to -90, we can't get a second action potential to occur immediately. So that dip there, that hyperpolarization is sort of like a pause in the process to give the sodium gates time to reset themselves and the potassium gates time to reset themselves, and the sodium potassium pump to quickly change the concentration of the ions again, so that we can do this whole thing again. It's sort of like a built-in pause for us, and we're talking like milliseconds. We're not aware of it. So that's the reason why we have that undershoot that takes place, where we go all the way down to -90. It just takes a little more effort on the graded potential to get us to the threshold. Last point. Not every depolarization event of a sensory receptor is going to trigger an action potential. I've been saying this all day now. The strength of the stimulus of the graded potential is dependent on, what are we being triggered by? What's irritating our sensory receptor? Odor, pressure, well, not odor, but pressure or pain or touch or temperature, anything like that. What's going to trigger the depolarization event? Is it going to be strong enough to hit threshold? If it doesn't hit threshold, then nothing happens. We either generate an action potential or we don't. And a lot of our sensory irritation that we are not aware of is because we don't hit threshold. So if someone says, "You smell that? It smells like rotten eggs," and you say, "No, I can't pick it up. I must not be able to hit threshold with that odor." That's what's happening. You can't smell the same thing that someone next to you can because you're not hitting threshold. So, okay. Yeah. We'll start here on Thursday. So this is a good place to stop. Anybody have any questions on any of this? Okay, let's get out of here. Lab this afternoon is going to be on the special senses, looking at the eye, the nose, and the mouth, taste buds, olfactory reception, and then the structures of the eye.